All right, so good afternoon, everyone. EST time. Welcome to today's seminar. Thank you all for taking the time to join us. My name is Diana Azam. I'm one of the board members of the Society for Functional Precision Medicine. And before we get started, I wanted to take a few minutes to share with you a couple of announcements. First, a reminder for all members to pay their annual dues for membership if you haven't already done that. It's important to keep the membership active. Memberships provide you with exclusive benefits. For example, it gives you access to recorded seminars like today's seminar, and also there are discounts on meetings that are are organized by this or part of the Society for Functional Precision Medicine. So I would really appreciate if you guys please try your best to keep your membership active and pay your dues. It's a great way for us to stay connected and also make the most of what our society has to offer. The second thing I wanted to say is that we're looking for individuals interested in joining our membership working group. This group will focus on developing a plan to increase memberships and in our society, and we really would love to hear your ideas and insights. So if you're interested to be part of this group, please don't hesitate to reach out to me directly. We have a couple of people already part of the group, but it would be great to have more additional members to help us in this important initiative. So thank you all for your continued support, and I really look forward to a productive seminar. Without further ado, I wanted to introduce our speakers for today. We are fortunate to have with us Dr. Venu Pillarisetty and Dr. Sheela Damle from Fred Hutch Cancer Center. They will be discussing tumor slice cultures as a window into the human TME. Dr. Venu Pillarisetty is a surgical oncologist and researcher dedicated to understanding the immune environment of pancreatic and other solid tumors. His laboratory focuses on exploring how the immune system interacts with cancer, aiming to develop more effective patient-centered therapies. Through collaborative research, Dr. Pillarisetty seeks to contribute to improving outcomes for those facing challenging cancers. Dr. Sheela Damle is a medical oncology fellow, immunologist, and a postdoctoral fellow in Dr. Pillarisetty's lab, who works to expand our understanding of immune cell-cell interaction in pancreatic cancer and the implications for anti-tumor immune responses. Through understanding of the immune microenvironment in pancreatic cancer, she hopes to drive better immunotherapies and discover novel treatment strategies. So I look forward to your talks. For those who have any questions, please feel free to enter your questions into the chat during the presentation, and then we'll address them during the Q&A session following the talks. So thank you all, and I look forward to a great presentation.
Okay. Well, great. Thank you so much, Dr. Azam, and thanks to the Society for the opportunity to present to you guys. It's really exciting. I've known Chris Kemp for a long time, and we're colleagues and have applied for grants together, and we're hoping that the NCI still exists and that the grant that was reasonably well scored will actually get funded, but you never know. Yeah, it's been quite the journey. But yeah, no, it's really exciting to speak with you all. This is kind of my entire research career is a passion project. In some ways, I don't have to do it. I could have a very comfortable life just doing surgery, and I still have to operate, so it's kind of harder to do both. But I think it's worthwhile because a lot of what we deal with in surgical oncology is diseases that surgery just does part of the job and we don't fix everything. So I want to talk a little bit just kind of broadly about, what I think of as the problems with cancer treatments in general across like all diseases. And this is probably the group where I least need to tell people that drug treatments based on animal models often fail in the clinic. Cures for lots of the diseases that we deal with, lots of cancers, are extremely rare, like true cures. Testing new therapies in patients, there's a lot of hurdles, obviously, to doing it, both ethical and then just pragmatic and really paperwork wise. And, there's a trial. I'm going to talk about a trial that we're very close to getting done that I spent 5 years trying to get it through the SWOG mechanism, NCI, and at the last level, it got killed because they thought there's not strong enough evidence to do it in quote unquote early stage disease. And I'll tell you why that's just crazy. We don't understand how a lot of treatments really work, so we don't understand how to deal with when they don't. And then immunotherapy, which I've been an immunologist for well over 20 years since I did my, kind of research fellowship as a resident. But it left a lot of diseases behind up to this point. So I think what I'm going to show you is reasons why we think we're going to get there, even in diseases people said immunotherapy doesn't work. So our lab's the tumor immune microenvironment lab. So the goals, trying to develop immunotherapy regimens to cure patients with previously uncurable diseases. My experience over the last 15 years on faculty has been trying to gain understanding of the tumor microenvironment of really difficult human cancers. Ultimately, about a decade ago, we realized we needed to create model systems that could tell us what was happening in the tumor microenvironment and be able to do experimentation, be able to test those potential therapies in vitro, and then ultimately, create clinical trials based on our lab findings. So I'm going to start with the understanding of tumor microenvironment. I'm going to focus on pancreatic cancer for this. We've studied a lot of different GI cancers, especially. But pancreatic cancer, I consider the ultimate problem. It's probably why I deal with it, because I guess I just like to suffer with my patients. It's really hard. And I'm a pancreas surgeon. I know when I operate on people that I'm not curing them. So long-term survival, if you took everybody, is still less than 10%. And then even patients with early stage disease who get surgery, chemotherapy, radiation therapy, they're typically going to die of that disease within a few years. So what we call early stage, and that was what I was alluding to when the GI steering committee didn't want us to do the trial on a earlier stage. How can you experiment on people with early stage disease? You're taking away their chance for cure. I would say they don't really have a chance for a cure. So we really need to do the trials in the places where we have data. Immune checkpoint inhibitor has not yet been effective, but there are reasons for that. And then targeted therapies are just available for less than 10% of patients because almost 90+% of the patients have KRAS mutations, and that's the main driver. So, and this is survival after surgery for early stage disease with or without chemotherapy. So this is without chemotherapy because this was published in 2007. Now, this is a really long follow-up. Like this is out to, like 20 years. But you can see most of what happens in that first 5 years, and the survival is really poor. This is a more updated study using the most common now standard chemotherapy for pancreatic cancer, modified FOLFIRINOX, versus gemcitabine, which was the previous regimen. This is only out to 5-year survival, but you can see this is like up at 50% 5-year survival. This is aspirational, as many clinical trials are. This is not really what we see even in practice. In reality, more than half the patients are dead at this point. And these are people who got surgery, had a pretty good stage of disease, and then had 6 months of multi-agent chemotherapy. So we need to do more. You could ask, well, are mouse models of human cancer accurate? And of course, in some ways they are, in some ways they aren't. This is actually a great paper. I don't mean to disparage them, but what this group did at Penn, they looked at the most commonly used mouse model of pancreatic cancer, which has KRAS, P53 mutations, all this stuff in it. They took cell lines, and some of the cell lines were ones when they put them in the mouse, they had lots of T cells, and some of the cell lines had small numbers of T cells. And the ones that had lots of T cells were responders to combined immunotherapy, and the ones with low T cells were not responders. Nothing super surprising there when you see it. They went back and figured out that there are things about the cancer cell that drove whether you got that T cell response or not. But what I wanted to point out was here is from a bunch of human tumors that they looked at, the percentage of the CD45 positive cells, so those are immune cells overall, the percentage of those that are CD8 positive T cells, basically sorted out into a bimodal distribution of ones where it was around 15%, give or take, or 30 something percent T cells. So lots of T cells versus not that many T cells. And then in the mouse, you see the same kind of pattern emerge. But what's a little sneaky is look at that y-axis. And I just wanted to show the scale of that, just for comic relief. This is human versus mouse in terms of T cells. So even the least infiltrated human T cell is, a T cell infiltrated pancreatic cancer has like as many T cells as the most infiltrated mouse tumors. Now, this actually fits with what we had previously published. We published this 10 years ago, where we did immunohistochemistry, did flow cytometry, found that there were a lot of both myeloid cells and T cells, the arrowheads, throughout pancreatic cancers. They tended to be traveling in the stroma, and a lot of times you'll hear people say that the T cells are stuck in the stroma or incapable, like they're not able to penetrate through it. But what we've observed, and we can say with direct observation, and Sheila's going to talk a little bit about that, that they're not physically stuck, but they are chemically potentially immobilized or constrained to that stromal area and not necessarily going right to the cancer cells, but they're only like 20 microns, 50 microns away. They're not far away. We looked and found that there were obviously this is in blood, PBMC versus tumor. This is the fraction of leukocytes. And then similar to what was found more recently, again, like populations of CD3 positive T cells in pancreatic cancer, well-differentiated cancers and poorly differentiated cancers, they're often a lot of T cells. And we did a lot of other studies that I'm not going to get into, but basically showed that they were functional, they were active, they could do something. So it would make you think that you must be able to do it. So, it kind of gave me this model of like how, how do we target this complex tumor microenvironment? At this point, a decade ago, we were just trying to understand it, but we had no way of testing anything. But at least what we were getting to was that, there's cancer cells, there's fibroblasts, there's macrophages, myeloid cells, and then T cells that are either maybe suppressive versus activating. And if you think about like each of these populations and kind of separate it out, and there's a long list, and I'm not going to go through everything, but like some of the things that we think about for like T cells, maybe we need to activate them better, or maybe we need to add more. If you get to the myeloid cells or macrophages, people have tried depleting them. Actually, that doesn't work, but maybe we need to repolarize them or get them to convert. We are starting to kind of believe that some of these cells that are the early myeloid cells in the tumors might be the precursors of dendritic cells in the tumor itself and not even require like lymph node trafficking for that to happen, and that some of this is happening like in situ. Stroma, can we alter the chemokines to allow the immune cells to move around? There's a lot of other things we can do. And then obviously, direct cytotoxic therapy, increase the susceptibility to immunotherapy. There's all these different ways that we can target it. And I think in the end, the smart job will be to kind of hit as many of those as possible. Now, bringing up one last thing, and this kind of comes back to why does immunotherapy failed initially? I told you a lot about the primary pancreatic cancers. That's what we were studying. It's actually pretty hard to get enough liver metastases to study. I don't know how they did this in Indiana. This paper just came out a couple of months ago. And obviously Nature Genetics, they did spatial transcriptomics of primary pancreatic cancers and pancreatic cancer liver metastases. And the top line information I want to give you from this is that the primary pancreatic cancers were like what I showed you, with lots of stroma and lots of immune cells and plenty of T cells, whereas the liver metastases were more cancer cells, fewer fibroblasts, less stroma, and fewer immune cells. And I think that provides some context for why all the trials that were done before in patients with metastatic disease that's mostly in the liver, that's another reason why those checkpoint therapy studies may not have worked. So I mentioned our goals, right? So this was to get a better understanding of the tumor microenvironment of human cancers, and then create model systems that accurately recapitulate the tumor microenvironment. So what we did, because we needed to figure out how to do experiments, right? Because I knew how to do experiments in mice, but how can we do something with these tumors? And I would say the one good thing about being, there's only one good thing I can say about being a surgeon scientist is that you have access to patients, and 99% of them are very happy to give you a piece of tumor once you've taken it out if you say, I'm trying to figure out how to cure diseases, right? So what we did is we figured out that we could take cores of tumor once it's come out. We use a little punch biopsy, 6 millimeter punch, take cores of tumor. We have the pathologist standing right there to make sure that they're happy with the margins and that we're not breaking any, College of Anatomic Pathology rules or anything like that. And then we get that to the lab, and we don't really use a razor blade, but we do use a vibrating microtome, and it's essentially an extremely expensive, extremely precise micro deli slicer. So you're taking your little slices of pepperoni or whatever and making uniform slices, putting them in a culture well, plus minus how you do it. There's a lot of different ways to do it. I don't really want to get into that kind of stuff, but and Sheela's going to talk a bit about the kinds of things that we're doing at this point. This is what we envisioned, all the different kinds of stuff we could do with it. We can take the fixed tissue, do immunohistochemistry. A lot of what we've done is just looking at cleaved caspase 3 staining or looking at the histology. We can look at TCR sequencing, so see whether T cells are clones or expanded, measuring cytokines from it, digesting the cells into single cell suspensions, doing flow. At this point now, single cell RNA sequencing and live imaging. And then on this IHC level, next level is spatial transcriptomics, which we've started to do some of that kind of stuff. And it's been pretty exciting. Just a little bit about those slices. This is what we published a while back now, but the kind of main points we were trying to make with this is they actually look pretty good, and they'll stay in culture histologically, look not too bad. Even after a little over a week in culture, the MTT assay is showing metabolic activity that it's kind of roughly about the same over time. It starts going down later on. The proteome, that's what's being seen here, comparing like day 6 in culture versus day 0, is overall relatively stable. The tumor slices the Ki67 rate, so proliferation is fairly similar after 6 days in 6 days in cultures, as is the cleaved caspase 3 percentage. So cells are actually growing and dying during this process, and they exist and are living maybe not their best lives. It's not as great as the cancer sitting in the patient, probably, for the cancer, but they are able to survive long enough for us to be able to get a snapshot of what's happening in them. We were very interested in immune cell populations, and fundamentally, they're really similar over that time too. There's probably some that are dying, some that are changing, and we've learned over time that there is an active response going on this whole time, even if you don't do any kind of treatment. So, we've been working on this for a while. We have like a bunch of publications, both looking at chemotherapy, immunotherapy, in pancreatic cancer, colorectal cancer, liver metastases, and really getting into a lot of like the biologic mechanisms behind how things are working in the tumor microenvironment. So I wanted to get to, testing the potential therapies in vitro. And one of the papers that even flashed up there, I'm going to let Sheela start talking about it because this has been kind of a launching point for a lot of her work in the lab. And I'm going to hand it over to you.
All right, thanks, Venu. Yeah, so
It's back.
there we go, it's
Yeah.
back.
Yeah.
Like Venu mentioned, that we want to use these models to try to understand what's going on with patients' tumors and be able to test different therapies. And, one of the first things that I had seen in the lab was using PD-1 blockade, so immune checkpoint blockade in the slice cultures. And we can see that in the tumor slices, when we treat with either control or anti-PD-1 monoclonal antibody, you get increase of the cleaved caspase 3 staining, this is by immunohistochemistry indicated by the brown staining here. And we can kind of use that as a model to try to think about how to test other things and how to build upon immunotherapy, which we know clinically doesn't work that great, but maybe there are things that we can do to it to kind of enhance it. And so before I joined the lab, they were investigating other combinations with PD-1 blockade, including chemokine inhibition. Venu mentioned that you can see a lot of T cells within the stroma of tumor, but they appear to not be able to access the tumor cells. So they kind of posited, like, is there a chemokine gradient that's keeping them restricted to the stromal compartment? And can we block that to enhance their movement toward the tumor cells? And so we took our pancreatic cancer tumor slice cultures, treated them with either the CXCR4 inhibitor AMD3100, anti-PD-1, or the combination of those therapies, and then looked at this marker of apoptosis, cleaved caspase-3 immunohistochemistry. And with the combination treatment of AMD-PD-1 or AMD and anti-PD-1, we see that there's a great increase in cleaved caspase-3 staining, as you can see in these representative immunohistochemistry images, with the brown staining being the cleaved caspase-3. We can also see that there are differences in the immune cells, which we would posit would be the case if we're inhibiting this chemokine receptor that we think is on a lot of T cells. But we see just all leukocytes increase into within the tumor slices after culture with this combination treatment, and even with anti-PD-1 treatment, which is consistent with some of the known mechanisms of action of PD-1 blockade, increasing proliferation of T cells. And this was all done by flow cytometry with the representative plots here. Like Venu mentioned, we can also do sequencing of the DNA in these slices and look for TCR clones that are expanded. And we see with each of the treatments, with the AMD3100 and the combination treatment, all the different T cell clones within the tumors are all expanded, and to the greatest degree with the combination therapy, suggesting that there's an active immune response that we're stimulating with this treatment. Further in support of that is when we measure cytokine production, specifically CD8 T cell effector cytokines, granzyme B and interferon gamma, we can see that those are much higher in the combination treatment than compared with the control or the AMD3100 alone. It's just more apoptosis, potentially CD8 T cell mediated apoptosis. When we look at the mechanism of how this happens, we mentioned that CXCR4 is a chemokine receptor, and it's involved in T cell migration, among other things. So we wanted to ask, are the T cells actually closer to the tumor cells? Did we do anything functional that's causing this? And when we look at the number of, or the percentage of CD8 T cells that are near an EpCAM positive tumor cell, the combination treatment, AMD and anti-PD-1, increases the number of percentage of CD8 T cells that are near those tumor cells. And those tumor cells that are near CD8 T cells also are positive for this SR-SPLCA, which is a fluorescent reagent that's a marker of early apoptosis. And the combination treatment has the greatest degree of this staining. We can see that AMD3100 treatment alone or in combination with anti-PD-1 increases the percentage of T cells that are near tumors when we do multiplex immunohistochemistry or immunofluorescence. We can measure the distance between the T cells and the tumor cells. And we see with AMD3100 and with AMD3100 anti-PD-1, the largest proportion are close to the tumor versus within the stroma. And here are some representative multiplex immunofluorescence images, looking at the stromal compartment in yellow with fibroblast activating protein, EpCAM, which is a tumor marker, and then CD8 for T cells. So with the control, you can see there's a large stromal compartment. There's a few green areas within that stroma, and then tumor cells on either side. With AMD3100, you sort of get a bit of a breakdown of where those T cells are located. They can be either near the stroma or near the tumor. Here in the anti-PD-1, the T cells are mostly within the stroma, the yellow area. And with the combination treatment, you kind of just see T cells everywhere, and there's not that much tumor cells and not that much stroma around. And that's kind of indicative of the apoptosis that we were seeing in other markers. But the T cells are a bit more free to go around where they want to. And we see that this migration and cytotoxicity happens really quick after treatment, which is consistent with sort of a cytokine-mediated effect where we're not waiting for transcription or translation to happen. It's really sort of a gradient effect. So the top panel is just a 90-minute time lapse of CD8 staining EpCAM for tumors and SR-FLCA, that early apoptosis marker. And you can see that this staining is very stable over about 90 minutes. And then when we add with our anti-PD-1 AMD3100, after 15 minutes, you can see that the green T cell is adjacent to a tumor cell, and there's a little bit of the apoptosis marker, but you can see that expansion of that blue staining over time, and that T cell is sort of surrounding that red tumor cell. And when we quantify that, we can see that the EpCAM or tumor positive cells that are near T cells have more of the SR-FLCA staining, whereas the tumor cells that are not near a CD8 T cell don't have that EpCAM staining, suggesting that the CD8 T cells near the tumor cells are leading to that apoptosis staining. When I joined the lab, so this is sort of the background of me joining the lab, I wanted to find additional ways to evaluate apoptosis in these tumor slices. The immunohistochemistry is with SR-FLCA and cleaved caspase 3 is a great way to visually look at it, but it's pretty labor intensive. And I wanted to find a way to look at multiple different slices from multiple different patients to try to help control for some of the inter-patient variability and intra-patient variability. And so the common pathway for both CD8 T cell mediated cytotoxicity and chemotherapy and radiation is the caspase, typically caspase 3 cascade. And I first wanted to think about what are other targets of caspase that we could use as markers other than cleaved caspase 3. And we came upon cytokeratin 18, which is expressed in a lot of epithelial cells. And on the right here, you can see this is staining, strong cytokeratin 18 staining in various cancer types. And the strongest staining is in epithelial cancers, including pancreatic cancer. And cells that are not derived from epithelial cells like glioma or melanoma or lymphoma have significantly less cytokeratin staining, suggesting it's common in epithelial markers. And cytokeratin is actually cleaved by multiple caspases, including caspase 3, and leads to a sort of novel neoepitope that can be detected in the cell culture supernatant by ELISA. So with our tumor slice culture model, we get our biopsies, we cut our little slices, we put them into culture, do our various treatments, and then we can take the supernatant over time and measure this caspase cleaved cytokeratin 18 to study the effects of different treatments. So in this top panel, this is pancreatic cancer treated with staurosporine, which is a chemotherapy type agent that's known to induce apoptosis or control. And in the control DMSO, you can see there's very little change in the cytokeratin 18 that's cleaved. But over time with treatment with staurosporine, you get a nice time-dependent increase in the caspase cleaved cytokeratin 18. We also compared it to another viability assay, just another way to try to check ourselves that this was actually going to be a good readout of apoptosis or at least cell death. And we see a nice negative correlation between the viable fraction as measured by real-time glow, which is a sort of a luminescent or fluorescent agent, with the cytokeratin cleaved caspase 18. And going back to some of our other data that I showed before with the caspase cleaved cytokeratin 18 and AMD 3100 and anti-PD-1 therapy, we see that the combination AMD 3100 and anti-PD-1 has the largest increase in caspase cleaved cytokeratin 18 compared to AMD or anti-PD-1 or control. And I think this graph also highlights some of the inter-patient variability that we see. And this assay is really helpful because we can measure it over time, so we can control for the patient's own kind of baseline expression or apoptosis level, as well as between different patients, making it a pretty reliable marker for apoptosis in the slice culture model. It also allows for a dynamic measurement, so we were able to measure it over time. And that kind of also brought us to a point of being able to look at these slices over time using multiplex immunofluorescence. And we got this really cool microscope that is a confocal that can do live imaging. It has an incubator in it. So we can put our slices into a dish, stain them with different markers. In this case, CD8 for T cells, pan-CK for tumor cells, CD4 for CD4 T cells, and dendritic cells, and look at these slices under the microscope with fluorescent markers and look at how they evolve over time. These are just two separate areas of one particular slice, and you can see there's lots of green tumor cells, and there's kind of a smattering of different blue, yellow, and red immune cells throughout. I think, again, speaking to the fact that there are lots of immune cells just present at baseline in these slices. And when we really focus on just the immune cells, you can see that between patients, there's kind of a lot of variability. In this panel, the blue is CD8, CD4 is in green, and CD11C is in red. And you can see in the top left and top right, there's a lot of different immune cells there. And these arrows, orange and purple arrows, are indicating immune cells that are coming in close contact, which is something that I'm particularly interested in and trying to understand what's happening in these contexts. So we can see that these are present in lots of different patients, so 4 different patients here, but that they're present in varying degrees, which really speaks to the ability to be able to do this in real time in multiple different patients in multiple different slices that will really give us a better understanding of different biologies that are going on in patients. And these cell-cell interactions, I think, have been historically very difficult to study with flow cytometry or other more static techniques. But I think with this microscope, we can really get pretty fine detail into these potential interactions. And so the staining colors, I apologize, are different again, but blue is CD4, FPM is a tumor marker, and green CD8 is a T cells, and CD11C are dendritic cells. But you can see there's what appears to be a dendritic cell in the middle. There's some small punctate blue CD4 staining, some yellow CD8 T cells around it, and then kind of little blebs of FPM green staining, which to me look a lot like an apoptotic cell. Obviously, this needs to be validated with kind of orthogonal approaches, but I think you can imagine watching this over time and watching this evolve, you can gain a lot of insight into the underlying biology of what's happening and what happens with different treatment. And I just wanted to share one more immunohistochemistry slide, kind of speaking to being able to understand things over time. This is a PDA tumor slice, several different slices treated with the similar treatments that I told you about before, their control, AMD 3100, the CXCR4 inhibitor, anti-PD-1, or a combination. And then we looked at different immune cell markers and tumor markers, and this is day 1 after treatment. You can see that in the IgG control in these 4 different tumors, they look sort of similar. There's some green staining, a little bit of blue, not a lot of red, not a lot of yellow. As we do different treatments with AMD 3100, as we saw before, there's a little bit more blue staining and a little bit more yellow. With the PD-1, maybe there's a little bit more blue, indicating some more CD8 T cell infiltration. And then the combination treatment, you see some one slice has a lot less green in it, and we think maybe there's more apoptosis going on. We have some evidence to show that before, but there's kind of differences in how much of different immune cells are actually being infiltrating. And so it's a way that we can look at things over time and look at a lot of different slices with a lot of different markers. And I think all of this is really to develop clinical trials based on our findings to be able to have robust ways in vitro to study new treatments and to understand the biology so that we can translate this into people. And I think I'll keep going, but
Talking about this trial.
yeah. So this is a trial that we're working on getting started, which is a randomized phase 2 study of neoadjuvant NALIRIFOX, which is a triplet chemotherapy, with or without cemiplimab, which is anti-PD-1 antibody, and motixafortide, which is CXCR4 inhibitor, in resectable and borderline resectable pancreatic adenocarcinoma. If you'll recall, Venu mentioned that he's been trying to get a trial similar to this in this early stage disease, but it wasn't able to happen. But we think that we can do it now as we have, I guess, more.
I mean, we just couldn't do it on a national, like NCI clinical trial level. So we actually had to then go to the drug companies, get money, and get some philanthropy money also. So we're going to run it ourselves internally. Which actually has its own advantages and will probably be more efficient for our science side of things. And we're running it like almost like a scientific experiment because we have the chemotherapy alone group. We've got the chemo plus the PD-1 inhibitor, and then we've got the CXCR4 and PD-1 plus chemo. So the science that's going to come out of it's going to be really cool. And we plan to do some slice cultures on the tumors as they come out as well. So this kind of pulls us to, where we think the future of this stuff, slice cultures in our lab. We're still going to use it as a platform for discovery and mechanistic studies. Definitely going to incorporate it into our clinical trials. And then one of the big things is integrating with other precision medicine techniques. So I alluded to earlier, Chris and I are collaborating on trying to use the platform that he developed that's part of Sengen to connect it where we get the same patient's tumors and port it out to those 2 completely different types of platforms. One that's really optimized for highly like testing large numbers of drugs that are focused around the cancer autonomous effects, and then one that's really complex and focused on the entire tumor microenvironment, and then being able to integrate those data into an actual overall platform. So that's kind of where we're hoping to take this in the long run. So thank you all for your time, and we have plenty of time for questions. So thanks. And I'll just stop that. I can always share if needed. Anything else? Sure.
So if you have any questions, feel free to unmute or throw them in the chat. And Diana can also field some as well.
Yeah, I thank you. This was amazing. I just wanted to ask a quick question because I have to log off. Is right now in your clinical trial, I mean, you're not really incorporating the tumor slice cultures as a way or as a tool to guide the treatment. And I know you just mentioned that this is something you guys want to do. I feel like why aren't you incorporating it now in this study? Like, what are the challenges and what is it that we need to do to start using these tools, right, to guide these arms or to really make to show that they can be clinically useful?
Yeah, there's a lot of challenges. I mean, you know, for using something for deciding on treatments, you'd need it validated to begin with, right? So we're not at that point. It's just difficult for what we're looking at, where you are looking at the overall tumor microenvironment. It is, you need a certain volume to feel comfortable that the immune response is reflective of what you're going to expect to see in a patient. So we're using large pieces of tumor, right? These are 6 mm cores. These are not the kind of pieces of tissue that you get from a percutaneous biopsy. These are surgical specimens. The trial that we're planning is a neoadjuvant trial, so it's before the tumor's out. Now, we are hoping to learn enough from this that in the future, if for example, patients are going to then get their tumor out and then go on immunotherapy or something, we might do that. There's more complexity to it because a lot of us also think that immunotherapy works best when you have the primary tumor in there, because everyone's heard of TIL therapy. Well, we're trying to essentially do endogenous TIL therapy without taking the tumor out. We're trying to activate those tumor infiltrating lymphocytes in the body before we remove the tumor. That said, we definitely want to move the scale down so that we can use smaller biopsies. So one of my colleagues who we share the same lab, Ray Young, he's been doing this work with me for the last almost decade. He's also a surgeon. He primarily does liver surgery, but he has a couple of big projects right now that are using just small cores to try to analyze them and see what you can learn about potential for response to therapeutics. I think, though, for immunotherapy, those really small ones are going to be difficult, because part of the aspect of immunotherapy is that you've got a lot of different T cell clones. You don't always know which one is the critical one. So unless you have a certain volume, you might not be able to do it. So sort of the short answer is, I do think we will get there, but that's not going to be like the quickest part. And that's why our first priority has been around like just, you know, using this to understand the biology so we can come up with better therapeutics. And the other interesting thing is that even though there's heterogeneity in response, I mean, the publication that we were referencing from 5 years ago, we've done that drug combination in pancreatic cancer samples. It's actually unusual for them not to respond and have an impressive response to that combination of drugs. So for the moment, we're quite hopeful that it'll actually do something. So it'll be to be seen.
Thank you.
Thanks. Appreciate that.
Hey, Venu. Hey, that was great.
Hey, Chris.
I
Thanks.
love the tone. Really nice. Thank you, both of you. Do you have any clues as to the genetic background of the patient or the tumor itself as influencing what you're looking at? So prominent mutations in DNA repair genes as comes to mind immediately, right, as an example.
Yeah, that's a good question. I don't know that I don't know if I've seen enough of, like, so, yeah, I don't know.
Yeah, we don't really know. I mean, it's super complex.
I guess if you ever saw like a really outlier response, you would want to dig into, is there a homologous recombination defect in that tumor, for example,
Yeah,
or mismatch repair or that sort of thing. Which would,
Yeah,
you'd see where I'm going.
yeah, definitely.
I mean, the thing is, we do see really good responses, but if we saw a really good response and there was a huge immune infiltrate, it's but so for example, like those kinds of tumors in pancreatic cancer probably represent like 1 or 2%
Yeah, it's very
of
low.
the super rare. So like almost not enough to be able to detect it in some ways, in our experimentation. So we might, we don't even know if we've seen any like that at this point. Yeah. We've probably done it like on 50 pancreas tumors or something, or, maybe 50 to 100, but I can't recall seeing something where like, oh, wow, that's so different. Yeah.
Uh-huh. Okay.
Great point. Yeah.
Okay. Maybe tomorrow.
I don't know. Yeah.
Yeah.
Do we have a tumor?
I
don't think there's one.
No
tomorrow,
pancreas
but-
tomorrow.
No pancreas tomorrow. Yeah.
Okay.
I mean, we are,
Okay.
we're held hostage to the OR schedule. So, whatever great experiments the people in the lab have, sometimes they'll have it planned for like a month and a half. For some reason, we don't have a certain type of tumor, and sometimes we'll have a whole bunch. So it's definitely harder than other types of research that involve like mice that you can order from Jack's in that
Yeah.
sense. Yeah.
Yeah.
Thank you.
I had a question. Hi, this is Annalise. I had a question about maybe within the same patient, like, did you take multiple slices from the same patient and just a little insight about the heterogeneity you may see just within intratumor specimens?
Yeah, great question. Yeah, we typically, when we do all of our treatments, we do them in at least 3 slices per patient. And we kind of, as you're slicing through a core that you've gotten through a biopsy, you can grossly see differences in the slices, just in terms of how much tumor there is, how much stroma there is. And so we do try to balance that between just the gross appearance between all of the treatment groups, and then do multiple slices per treatment group. And then imaging multiple areas within the tumor. But you definitely see heterogeneity I think most prominently in the stroma and tumor content. The immune cells are kind of there or not there. There are occasionally some pockets within tumors, but I think within each patient, I would say that there's kind of the immune cells that you're going to get. And it's, you don't typically see one slice from a patient that has tons of immune cells and then the next slice has none. I would say that is actually fairly uniform.
Thank you.
Yeah, thanks. Good question.
All right, Bruce had a question next. Bruce, go ahead.
Yeah, thanks for the presentation. I was looking at, I'm on Fred Hutch's Cancer Center, looking at all the labs that you can search through. I was trying to find your lab. I see this one by this Kemp lab for functional precision medicine, but I couldn't find the TIME lab.
It's University of Washington.
Yeah.
Okay, University of Washington. Here we go. Thank you.
Yeah. Yeah. If you just look up my name, that's the thing that's gonna find it. But we're not great on websites, I guess.
Thanks.
Not
I was looking in the wrong place. Thank you.
Yeah,
Yep.
no problem. And I don't, Michael Bergman.
Yeah, Michael Bergman's next. Go
Yeah.
ahead.
So thanks for the presentation. We also do slice cultures for 5 years, and we are very much interested in the T-cell response. So what's the time frame you use? I mean, you told us the data after 3 days. So what did you look at 5 or 7 days, whether you see something different?
Yeah, so I think we know that the immune cells stay viable in the cultures for over a week. I think based on the cleave caspase 3 staining and the caspase cleave cytokeratin 18 staining, we really see a maximal effect by 3 days. So it's very fast. We have looked at day 6, but you get a lot of apoptosis and just really fatty looking tumors by that time.
The reality is like what we did for about the first 5-6 years was mostly day 6. I mean, we really were looking a lot at that because that was where we could see the endpoint most easily of like what was killed. Over time, we've been moving our study of it to much earlier time points. And at this point, we're now looking at like immediate, like we take it out and we're treating and putting it on the microscope now that we've I mean, we literally got this, we've had access to confocal microscopy for a long time, but we just got our own one a couple of months ago. So now we're just trying to look right away.
Yeah, and
Lots happening.
yeah, and we can see, you see a lot of movement within the immune cells right out of the patient. And so we think that that is actually a relevant time point. I think, we see that, the T cells migrate toward, tumor cells early within hours, and start inducing that apoptosis cascade if they're primed to do so, or induced to do so with treatments.
Yeah, and actually it really depends on the tumor and depends on the treatment. So we spent all this time talking about pancreatic cancer, but we've done a lot of work on colorectal cancer, liver mets. And as I mentioned, that publication on pancreatic cancer, liver mets, basically colorectal cancer, liver mets are kind of similar. They're relatively devoid of immune cells. They don't have that much stroma. But we found that if you block IL-10, that you wind up with proliferation of immune cells and anti-tumor activity, but that takes longer because I think it's creating somewhat of a de novo immune response, or cells that are not quite as primed. So it takes several days for the activity to happen there. Whereas for this CXCR4 PD-1 in a pancreatic cancer where there's a ton of effector T cells already there, you can see a response like in an hour. You'll start seeing the cells moving and killing.
And if I may ask, did you look at, did you do FAC staining of myeloid cells, of macrophages, like a little on day 3 or day 4, because they are maybe the most vulnerable in those in vitro systems?
Yeah, that's a great point. Yeah, we have done FAC staining on those, and you can see some drop off in the myeloid compartment at like day 3, more so at day 5 and 6. But I think, I think FAC staining of myeloid cells is
hard.
And
well, the IHC that we did basically showed stability of the different populations, including myeloid cells. And then we've seen more recently with the activation that we're seeing like higher numbers of dendritic cells even after a few days with the treatments.
Yeah, so I think that they're able to maintain within the cultures and actually do some functional things there. But I think anytime you're going to chop up a tumor and stick it under a flow cytometer, you're going to damage the cells, and the myeloid cells are particularly susceptible to that as well.
Yeah. The tumor type matters a lot. High stromal content tumors tend to do very well in the slice culture. I don't know what kind you work with, but the ones that are high vascularity, softer tumors don't do so well.
All right, next we have Colin Paul.
Yeah, hi. Thanks very much for the presentation. Any thoughts on, you tease this a little bit, different types of studies or where you would choose if a traditional organoid approach versus the slice culture approach, whether some of these immunotherapies are even possible with the organoid methods where you're digesting things more, and maybe some of that data is to come, but just curious your perspective on relative advantages of each of those.
I actually had a slide on that and I took it out because I wasn't sure how to represent which was better than what. But because I was thinking about it from this like cascade of complexity to like simplicity versus complexity, and then like how representative that might be of like the overall environment. But I guess my simple way of thinking about it is, and as I was mentioning in that work that Chris and I are doing, is that for something like organoid testing, I see that as ideal for where you're screening large numbers of drugs and trying to look for vulnerabilities on the cancer cells themselves that you didn't, that you might be trying to discover for a specific patient's tumor. Whereas, to me, it wouldn't make sense for it to be immunotherapy unless you've got the immune cells. It's really difficult to expand the immune cells to the point that you can then think that those are fully reflective of how they're going to work normally. So if you take TIL and you now expand them out and then you add them into an organoid, I don't know if whatever drug you're using to activate them is the thing that worked, or is it because you threw the cells were bathed in IL-2 and IL-15 and various other things. So to me, it's more the things that require like complex immune environment interaction, stromal cell, things like that is where something like a slice culture is particularly valuable. As far as a personalized therapy kind of thing. And then stuff where you expect this is going to kill the cancer cells, seems like something like an organoid type testing would be the thing that makes sense. But I don't know.
No, that makes perfect sense. The other aspect, Venu and Colin, is of course, you have to have surgery as part of the patient's treatment. Access to tissue is obviously a thing. And then where you can think of it, this and also tumors where there is no available therapy. So if tumors have 3 lines of standard of care, it's unlikely you're going to be able to convince a doctor to try something new right off the bat. So tumors where there's a dire need for any kind of targeted therapy or any kind of new approach. So these are all really interesting things to talk about, and the SFPM is a great venue to do this. So a lot of us don't have to make the same mistakes as we try to do this. We can learn from each other. So again, the SFPM is a venue to stimulate this kind of thing. So I encourage anyone who is listening to get in touch with Venu or myself to work together in the future. But great question and worth a lot of. We could talk about that for quite a while. But thank you, Colin. I think it was Colin.
Yeah, and yeah, as Chris said, I mean, we're happy to speak with anyone. And Michael, you asked some questions, didn't say you do slice culture. Happy to chat if you have questions, or we certainly could probably learn from you too. So appreciate that. Thank you.
Great. Thank you all.
Thanks.
All right,
I think
well,
did we lose
yeah.
our host?
She had to leave, but I think if we have no more questions, we can adjourn about 6 minutes early. Everyone have a wonderful holiday, and our next seminar will be in January on Wednesday, January 8th, same time, 12 p.m. Eastern. Christine Cruz and June Yang from St. Jude's will be speaking. Hope you all can make it.
See you. Thank you.
Well, thank you all so much. We appreciate the ability to speak with you guys. Thank you.
Thank you very much.